And welcome to the keynote session for this meeting. I'm very excited about this. We have speaking to us Dr. Tony Mire, and it is my honor and pleasure to introduce him. Tony is presently the head of global quality at AstraZeneca. He is trained in genetics and biometry and has a PhD in cell biology and biochemistry. I have had the misfortune, pleasure, and privilege of knowing Tony for more than 25 years. I met him while he was still a scientist at the National Institute for Biological Standards and Control in Potters Bar, UK. There he was the head of the cytokine group in the Division of Immunobiology, specializing in development of assays for characterization and quantification of biological products. And Tony and I were part of a cadre of young and upcoming scientists that were pushing on the heretical idea of surrogate assays being used for bioassay potency assays, which was now it's kind of par for the course. While I was working at Genentech in the 90s, I was part of a team that actually talked Tony into giving up his really posh life as a regulator in the UK to come across the pond and try his luck in the States. He became the director of bioanalytical sciences at Genentech. And one of the first things he did with his newfound cash is to buy a typical big American car, a big blue Trans Am, and practice his technique as a culinary master, which he still practices to this day. And his motto is that if it cannot be microwaved, it's not worth eating. After his time at Genentech, he then moved to a position as executive director of analytical sciences at CancerVax. That was in San Diego. After several years there, he decided to dip his toes back into the regulatory waters and moved over to FDA, where he worked as a principal advisor for regulatory science and review, Office of Biotech Products in Cedar, and head of analytical sciences and standards in the Office of Director at CBER, FDA. He was there for a couple of years, and then he moved back to the biopharma side, where he worked as a vice president for the North America contract, Singapore, and product quality at Amgen. He filled a lot of roles there. And he was in Boulder, he was in Thousand Oaks, he was all over the place, essentially staying one step ahead of the creditors, but I think he was pretty successful in that. Importantly, Tony, having had his feet on both sides of the stream in terms of regulatory, being on the client side and on the regulatory side, has a good feel for both what the agencies are asking for and the cost and challenge that brings to the biopharmas and trying to meet that in a scientifically valid fashion. Again, Tony's been at AstraZeneca now for several years, and he's going to present to us today on bioassay evolution. Where are we now? Tony, take it away, please.
Great, thank you, Mike. It's been a pleasure to know Mike for a few years in our history, and I want to thank everybody for attending this presentation. When I saw that I was a keynote at the end, I was hoping it's leave the best to last, or I just got shoved at the end for the sake of it. So hopefully you will find this talk interesting, because I think I was attempting to summarize a lot of the thoughts that folks such as myself and Mike and Jill have had to tackle over the years, and to really see where are we now with how we're dealing with bioassays. So a bit of a retrospective, and then trying to pull that through to some of the ideas and technologies that we're using at present day. So I always like saying, why are we still having discussions on bioassays? I mean, they were developed a long, long time ago. You know, when biologicals were first thought of, when, you know, insulins were first discovered. But what is interesting is, even after all this time, if you try to find any decent FDA or European publication from the regulatory side of exactly what bioassays should be, how you develop them, how you analyze them, how you deal with the results, etc. There's really a paucity of that type of direct regulatory guidance out there. There's lots of papers that actually people like, such as myself and Mike and Jill and others have published, but not directly from the regulators. Interestingly, even to this day, the way you analyze bioassays can still get questioned by the agency. We made huge strides, for instance, with the USP chapters on statistical analysis of bioassays, but it is amazing how questions still arise between statisticians on the best way to do things. There are new ideas popping up, not as many as I thought over the years when I look at what we consider, at least in my book, to be reasonably new. There hasn't been a ton of them, which is kind of interesting, but I'll get over and talk about those a bit later in the presentation. And then some technology advances that have occurred. But again, I wouldn't say that there's been revolution in our space, again, which I think is kind of interesting. So what are those eternal questions that I believe we still get when we are working with a new product, putting a filing together, and then working our way through commercialization and moving from development into the QC labs? Now, the biggest question always is what actually is a bioassay? There's been multiple definitions of bioassays over the years, and even the confusion of what's a potency assay versus a biological assay. And I'll have a little chat about that. People still question, oh my God, we've moved into the era of being able to heavily characterize, for instance, protein-based products, such as monoclonal antibodies. Why do we still need a potency assay? So there's still those questions that industry can tend to have. How close to being a mimic of clinical efficacy? If you're going to have a bioassay, the question always is, should it mimic what's actually going on in the clinic, and how close does that actually have to be? And do you even know the mechanism of action and how it works in the human? What format to use? There's many different formats from the old days that Mike was talking about, where people were using animals or tissues, to then cell lines, and then cells from tissues, and then cell lines themselves, and now immunoassays, and to all sorts of binding assays that you can do, biocore, etc. So which ones of those do you do and when? The classic issue with bioassays, because you're dealing with living things often, is assay variability. How do you reduce that? I think over the years, we really have got much, much better at understanding sources of variability and how to fix them. You then have, as I mentioned, calculating and reporting potency is still a question that a lot of folks new in the area have to deal with. And it still can be quite tricky, as well as how do you, as you develop during commercialization, moving from, for instance, a cell-based assay to receptor binding. So let's get into why do we need potency assays. And this is something that we've talked about a lot in the regulatory space, is that really potency assays are intended to be actually functional. It shows that your molecule that you're working with actually does something. You can have surrogates for that, but at some point during development, you want to show that the molecule you have actually physically does something, rather than just characterizing its structure and going, well, the structure looks all right, therefore it must be able to do what it needs to do. And if you're going to have a molecule such as an erythropoietin, or a molecule where glycosylation is heavily impacting PK and bioavailability, unfortunately, you're going to have to try some level of in vivo assays to show how glycosylation and different variants of your molecule actually impact PK and bioavailability. Only once you can do that can you then link the chemical structures to what you actually have happening in vivo, and then maybe move away from in vivo assays. And of course, if you're going to be doing immunogenicity assays and you want to show, do you have a sample that can neutralize the activity of your product, you're likely going to have to use some form of potency assay or bioassay to actually show that. You can show it's binding in a binding assay, but it doesn't necessarily mean it stops your product from working. So we do know what we want from a potency assay. So it's got to be biologically relevant. It's got to be linked to something the molecule actually does. You'd prefer it to be stability indicating, because you want to see if the molecule's physicochemical structure is changing, does that actually have any relevance to the bioassay and to its potency? And you want appropriate precision. You don't want an assay that's plus or minus 200%, where, quite frankly, you can't really tell whether the molecule's varying from lot to lot or going off on stability studies. And at some point, you want a bioassay that you've actually got to run in QC. So if it's going to be in QC, you don't want it to constantly fail because of some technological issues. So you want, as best as you can, a simple format. You want stable reagents. So, as I say, you want to go away from the in vivo stuff. You want to go away from tissues. You want to have, or bone marrow cells, primary cells. You want to get to cell lines that are more stable. You want to make sure you've got the proper control so you can see how your assay is performing. Is it varying from assay to assay, analyst to analyst, and day to day? You want it to be robust. You don't want it to be performing well one day and collapsing the next. So you want to have something that you can rely on. And at the end of the day, it's a regulatory requirement that assays, particularly for lot release, have to be validated. So it's got to be a validatable assay and everything that's associated with that. So when does a potency assay stop being a biological assay? And as Mike said at the beginning, when we first started in this business, we were in the world of in vivo assays, tissue-based assays, that sort of thing. So very close to, well, as close to the patient as you could possibly get, even though it's, it was mainly animal-based. And as time goes on, and you can change and understand your product better and better, you can start developing what we call those surrogate assays. So often during development, you will move through various stages depending on your product, in a way going from those much more in vivo type assays to eventually replacing your potency assay with either a direct ligand binding assay or eventually physicochemical tests. Some molecules have actually made it to physical chemical tests, but still in the biologicals world, most assays are still a biological assay per se. So how close to clinical efficacy should your potency assay actually be? And this is the struggle, especially with novel molecules and the cell and gene therapy space that we're moving into, and more complicated, even antibodies that have dual purposes and bispecific antibodies, etc. So if you look at what guidance is out there, it actually does say that mimicking the biological activity in the clinical situation is not always necessary, but it says a correlation between your clinical response and the biological assay should be established. So you have to think very early on, actually, in development, what is actually happening in a patient and how can I link that to the biological potency assay I want to use? And the reason why I say you've got to think about that early on is it can take years to actually get that understanding. And there's actually been times where molecules have been on the market for many years, and all of a sudden we discovered something around what happens in vivo that then has to be translated back to a novel biological assay that you had to come up with, even when the product's been on the market for some time. So we have that bioassay conundrum, and here's a nice pictorial that I've duplicated from Fred that he presented at a CAS meeting that kind of gives you the idea of how the relationship to what actually goes on in a clinical study, which is up the left-hand side, to how easy it is to do it in a QC suitability. And as you can imagine, in vivo models can be highly variable. You're using animals. It's not easy to do these for lot release. But as you go more and more towards analytical methods that look at the structure, in theory, you're getting more and more away from that direct relation to what's actually happening in a patient. So it's how far down that spectrum do you go and when during assay development, product development, and during the product life cycle. So one of the things that, and I mentioned at the beginning, that we always struggle with any form of biological based assay is variability. And there's lots of information out there now, luckily, because over the years, we've spent a lot of time reducing assay variability as best as we can to make these assays as reproducible and useful that we want in that QC environment. So I'm not going to go in huge detail, but we really do, hopefully by now, after all these years, understand the multiple sources of variability and of course do something about it. So we understand that cell lines can go off over time. So we have controls around how do you store them, how long can you grow them for, how do you feed them, etc. As you dilute your probably very concentrated and potent molecule into a bioassay, you may be diluting 1,000-fold or 10,000-fold into the bioassay itself. And that's one of the greatest sources of dilution. And it's amazing to me how sometimes you have to explain to people that doing 1 microliter into a mil is probably not a good thing to do. Is you need to be rational and have, you know, maybe 10 microliters into 100 microliters, or best 100 microliters into a mil, simply because of the capability of the pipettes you use. Position effects on plates is very important. We know that the outer wells of plates, when you put them in to the incubator, can have a huge impact to your assay. And even plate to plate, you know, your assay may take you 20, 30 minutes to set up. Well, once you've done the first plate and then got to the last plate, the plates could have been sitting out for half an hour, and you can get variability that way. How they even stack on top of each other can cause plate-to-plate variability. The reagents you use can cause assay variability, particularly if you change reagents over time, different buffers, different monoclonal antibodies. If you're doing ELISAs, they can change an assay. Your equipment has to be constantly calibrated to make sure that doesn't change over time. People are a big source of variability. Not everybody does everything the same way. And of course, as you move from laboratory to laboratory, it's not surprising that you're going to see some level of variation. So what are some of the things that have happened over the years? And it kind of seems a bit, wow, you know, really? You didn't do this all those years ago? Well, no, we didn't have a lot of automation in the early days. But now automation is becoming more and more the way to go, especially in the QC lab. And it has multiple advantages. Really, it gets rid of the actual dilution effect of diluting continuously, because the machine can do that much more accurately than a human can. But even there, humans do tend to get tired. If you're doing an assay where you've got multiple plates, we all get tired. And the pressure that you put on a pipette when you're dispensing stuff can be different from the beginning of the assay to the end. And of course, analysts themselves can have some bias around how they mix reagents, how they do it, how they use their pipettes, whether there's any drips at the end, etc. A machine removes all that because the machine does it the same way all the way through. And therefore, hopefully it can be more accurate because it has the ability to continuously supply accurately the same volume into your assay. And of course, some people tend to forget that, but there's a big safety issue. You know, I used to do a lot of bioassays myself, and boy, does your hand get tired after you've done multiple pipetting. And again, do a lot of that stuff, and you can end up with carpal tunnel syndrome and stuff like that. So having machines do it is much better. And of course, increased throughput. Machines can do a lot more plates in one go than humans can. But I do have a line there saying automation alone is not going to fix a bad assay. If you haven't optimized your assay and really looked at all those sources of variability, it can only get you so far. But look what happens with those graphs, just some data about invalid rates when you move from pre-automation to post-automation. And then look at the relative potency slide graph at the bottom on the right, where you have the variability of the assay that can drop tremendously when you're moving from a lab that only had humans doing it versus moving to automation. And it was much easier to transfer an assay that's automated than it is when you're doing it through humans. One of the things that was, at the time I was at Genentech with Jill, that came up that was very exciting was this use of the Echo auto diluter, where it's much, much more accurate than even doing pipetting with an automated diluting system that you saw on the last slide. It was incredible. And in fact, some of the assays that Jill's team were producing, you'd look at the curves and they overlaid themselves so well that you kind of went, did we actually put more than one sample in here? Because they were so accurate and overlaying themselves, which is just amazing, really. So that is probably an area where we have got much better at being able to get rid of these dilution errors that lead to the variability that we can see in assays that drive then assay invalids. And frozen cells, again, you know, we used to grow cell lines continuously. First of all, you know, you always had to have people coming into the lab all the time to maintain the cells, keep them growing, you know, measure the numbers, make sure that you had new media and all that sort of stuff. And as I say, eventually, with continuous culture, they could lose the receptor levels on their surfaces. All sorts of things can happen, where all of a sudden your assay would collapse and you wouldn't know why until you then looked at the cells and saw something had happened to them. So having frozen cells where basically you grow a huge bank of cells to a point in their cell cycle where you know they're going to have an optimal response, and then freeze them all down. And what you do when you want to set up the assay is you literally take the vial out of the cryo vessel, thaw the cells, add them directly into your assay. They wake up and they grow, and you get your result there and then without having to continuously having to grow them. Sounds like, well, you know, don't they all die and doesn't take them a while to grow up? It is amazing that actually cells can be very robust, particularly if you do a good job with the way you freeze them down. So very convenient, any time of day, get your cells out of the cryo freezer, get them up. Cells are never out of range because you know what you put in, you know that they're in a good source of or good state in their life cycle. Very consistent because you've got this huge bank of cells that are all in the same phase, that are all of the same age. So very consistent for results. Helps the method transfers because you don't have to have growing cells that you have to transfer to somebody. You just send them the frozen vials. And because each bank is qualified prior to use, you always have a source of predictable cells to use. Now, it also can save cost because you're not continuously feeding cells the whole time. You grow them up once and then you're done. And we worked out that it costs 1/7 of continuous culture if one site does your cell banking for you. So actually good business sense as well. And you can see the data. It is amazing how you can reduce the variability by looking at the use of the ready-to-plate cells. So not only you see you can halve the amount of variation you get, once you actually reduce variation, you can change the number of assays that you require to report a result because a single plate or a single assay is so much more reliable and precise that, again, it saves you in cost, it saves you in time. So although you have to redo your assay when you're moving from continuous cells to ready-to-plate cells for revalidation, that becomes part of your filing to justify the change. You file with the regulators and say, look, actually moving to frozen cells has made this assay much better, which helps speed through assay review with the regulators. And also using design of experiments. You know, with assays, it took a little longer for quality by design to hit how we actually develop assays. You know, originally it was very much on manufacturing and processing and looking at how to optimize production. But all the principles of quality by design work for assays as well. And a lot of companies have used design of experiments to optimize their assays much quicker and much easier than we would have done in the past, where you may have tended to do everything in series. Now you can do them all in one go, and you know, with more available statistics and more complex computational analysis, you can look at multiple different potential impacts to your assay to come up with the best way of doing things. And you can see here, just as an example, 3 different things that you would have in your assay if you have a coating of an antibody and you add cells to that, and then of course a time of how long do you incubate things for. With design of experiments, you can manipulate all 3 of those and end up with the best parameters that you can choose. Now, what is interesting, and it really gets to the analysis piece, is how do you control your bioassay and how do you actually measure and analyze the results? And you think after all these years, we've got it all down and everything's great, but it's amazing how people still can get questions from the agency around how are you controlling your assay, what parts of your standard curve, and do you have a standard curve, and what does it look like, what pieces are you using for analysis, how are you doing your analysis, and how do you make sure from assay to assay that your assay is actually being reliable. So you can see here the multitude of things, if you just take a standard curve, that you could be measuring. The lower asymptote, the upper one, the slope of the assay itself. You can look at IC50s or ED50s. You can look at the background. You can look at the maximal response. All these things are things you could use to analyze to say, well, how is my assay doing? So really trying to understand that is not trivial. And I always say you've got to have a really good relationship between the bioassay developer and your statistician to make sure that you're developing your assay in the right way and picking the right things to look at. So you'll get questions of, well, what happens if your slope does move up or down? What impact does that have in your assay response? And it's amazing, again, how sometimes people who are developing assays forget to look at that sort of thing. And so over the years, we've actually designed experiments to look at the impact of your slope, varying from a very shallow slope to a much stronger slope. And you'd say, is there a difference if I'm doing my potency assay results, whether my slope moves at all, whether it moves up or down or stays the same? Because again, from day to day, from assay to assay, a slope can change. So you actually have to do those assays to find out and look at the results. And again, feed that into the dossier that you send to the agency to justify how your assay looks under varying conditions. And so here's an example where we actively change the fold stimulation of the assay, so the up and down piece. Because again, you can have an assay that one day from minimal to max could be small or really big. Does that impact the actual relative potency answer that you get? Well, what we showed by moving the fold stimulation of the assay through manipulating the reagents and the cells, that doesn't really matter actually. If your assay is well designed and it's actually got a decent slope, the minimum and max really doesn't impact your ability to get a decent and accurate result. And it didn't really matter whether you were using two completely different types of analysis for the slope. There's different ways, as I say, when statisticians can use a 4P fit or parallel line analysis. Again, you could show the agency, doesn't really matter whether I could use one or the other, the results looked very much the same. So again, what is the impact of day-to-day variability on your assay? And as I say, regulators do like to see that you've actually investigated it. And of course, there's always that question around, ooh, this parallel line thing and comparing to a standard. Shouldn't you be looking directly in the assay itself and looking at your ED50s? And it never amazes me that that question keeps popping up. It's popped up for 30 years and it's probably not going to go away. But again, you have to show the regulators, you know what, if I pick that midpoint of the assay, yes, I can get a potency answer, but look at the variability compared to relative potency. And relative potency, as you saw from those original graphs, was comparing your sample to a reference standard. It's what's recommended by the World Health Organization. It's what's recommended by the pharmacopoeias. But it's amazing how people still get the question of, ooh, isn't that kind of cheating because it allows for day-to-day variability? But that's the whole point. If your assay shifts a bit, well, your reference standard shifts at the same pace as your product itself. And so it's much more accurate and precise using relative potency than it is just picking a point in the middle of your standard curve and fixing towards that. So hopefully, again, by showing this to a regulator, it proves to them that what you're doing is the most appropriate way of having a decent assay. So we talked about that cascade of assays over time, is how do you replace a cell-based bioassay if you want to? Now, I always say, if you have a really robust, well-characterized, well-designed cell-based assay, you have very few invalids, you get really reliable results, and it doesn't cost that much to do them. Now, you do need to have probably, aseptic processing facilities like a fume hood and all that sort of stuff, a sterile hood, and incubators. But at the end of the day, you've got to think about, is it worth replacing it? But if you do, there's quite a lot of work. You're going to have to persuade a regulator that moving away from a cell-based assay to maybe to a binding assay, that the two are going to be correlating between them. So you have to pick the types of samples you need. And again, we need to get better at knowing what sort of samples do you need to do. Well, you need to have some stress samples, because it's not just great to show, oh, you know what, my lot release assay, my lots look all the same. You want to say, well, you know what, if I did stress the molecule a bit, would my bioassay even pick it up compared to the cell-based assay? So you have to put in a variety of different lots, as well as different accelerated and stress samples into the two assays to see. You want to show that if you have a molecular variant, for instance, an oxidized product, will the oxidized product that maybe has a different potency come up with the same answer in a cell-based assay versus a binding one? Because if it doesn't, then what do you do? Because you may end up with a product that gets oxidized over time, that the potency assay would pick up the potency going down, but the binding assay wouldn't. So you need to, again, produce different types of variants, molecular variants, and put them in the assay. And of course, you want to have equal or better accuracy, precision, and reproducibility. It's going to be a tough sell to an agency to say, oh, you know what? I've replaced my cell-based assay with a, say, an ELISA-based assay, but actually it didn't have the equal accuracy or precision. They're not going to go for that. So you want to make sure, for instance, if you're working with a mAb, a monoclonal antibody, is binding the only part of the monoclonal's mechanism of action? Because if you are going to have glycosylation impacting the potency of the molecule, then you're probably going to have some assay that's going to have to measure it. It can't just be a binding assay. And of course, if you have, for instance, a FAB part of your molecule, you want to go, is it going to do the same thing from a binding to the ligand as a whole molecule would have done? And again, how neutralizing could that FAB portion be? So here's just an example of lot release assays for a product. And this was a product that's been around for, well, it's now been around for almost 35 years. And of course, you do lot release assay and you compare it to a standard, and you can see it's units down the side. And it does wobble up and down over time as you're doing lot release. Well, was the actual potency of that molecule and those lots varying over time? We just decided to, out of interest, to have a look. So we took, and this was literally 10 years of different batches. So this spans a 10-year production time frame where we had stored material, put them all into a single assay with a very good analyst who reduced the variability of the assay. And look, they're practically the same. So again, it kind of gives a bit of an indicator to the agency. And I love this graph is, you know what? Actually, the potency of our product really has never changed over time. That the only variability we're getting from lot to lot on lot release is the variability of the assay itself. And as we move with more and more of that knowledge and understanding of your product is, do you always need to have potency testing at the drug substance stage and at the drug product stage? And again, this is where data speaks volumes. Because if you can show that all you're doing is taking your drug substance and either filling it directly into a vial or a syringe, or diluting it and putting it into a vial and syringe, that actual extra manufacturing has no impact on potency, and you can produce enough data to show that, why would you need to have drug product testing on potency assay and drug product testing? Or vice versa, as an industry, we could say, or a company, you could say, you know what, we're not going to test potency on drug substance because we understand our molecule, it's frozen, it's stable. We understand that when we dilute it into drug product, everything's going to be fine. So we're just going to do drug product potency and take the risks. That risk is not patient risk, it's company risk. Because of course, if the potency is not correct at drug product that you could have predicted with drug substance, the company is the one that loses out, not that you're going to put subpotent material into patients. And this has been presented to the agency with appropriate justifications alongside physicochemical analysis. And several companies have now been successful to actually remove potency of either drug substance or drug product. And usually it's associated with some sort of physicochemical predictor of potency. And not surprisingly, a lot of physicochemical assays can be early indicators because they are more accurate and more precise than a bioassay. As you just see an example here of how size exclusion, if you look, for instance, on the right-hand panel, how size exclusion predicts the degradation much more in excess, but also actually if you looked at it from a time basis, it would show it quicker than the various assays, the bioassays that those three colored lines have there. And again, that's all data you can use with the agency to say, well, you know what, maybe for these types of physicochemical changes that we see through maybe heat, that actually size exclusion is a better predictor of potency going off than the actual bioassay itself. And you can use orthogonal methods to understand which ones of your physicochemical assays and which variants could go off prior to potency going off and which ones are actually important for potency. So you can control these through these orthogonal assays. And what we've actually shown that through DS drug substance release data, that having no OS results ever, whether you actually still need to keep measuring potency, where actually size exclusion and other types of assays can actually be more predictive of changes in potency than the potency assay itself. And here's another example of using data over time and experience is frozen drug substance. There's no change of potency, multiple batches over 3 years, nothing changes. So what is the point in continuously assaying potency of frozen drug substance? If you make a change to your process, again, you may want to be looking at the potency of material on accelerated stability or at 2 to 8, but there's no point in looking at it in the frozen condition because you're just not going to see any change. So in conclusion, hopefully that's given you a little history tour of all the different things that we've worked on over the years and where we are now. Is biological assays are still the only assays that can actually tell you if something is functional and active for biological therapeutics. The selection of your potency assay for lot release requires probably more than one assay to be looking at during drug development. You may go from in vivo to cell line to receptor binding as you go through drug development to prove that what you have at the end still correlates back to some of those more biological assays you started with. And the design of bioassays is absolutely essential if the results are going to be useful. If you don't spend your time designing your bioassay to remove variability and bias, then they basically have no value. So I hope you found that useful, and I will turn over to Mike and Jill for any questions. And I'd like to acknowledge some of the people that I've worked with over the years, including Mike and Jill and others. I really stuck with the Amgen team because Genentech was so far back, but Mike, you're in my heart. And this is based on a lot of people's work that you've seen during this meeting. So thank you.
Thank you very much, Tony. Thank you. I hope everybody enjoyed that as much as I did. That was an excellent talk, very informative. Jill, you want to handle the questions.
Yes, absolutely. Yep, we have a couple minutes for questions. So hi, I'm Jill Kraus-Andadini, and I'm going to help moderate the question session for Tony. So once again, thanks so much, Tony, for kind of that retrospective and prospective talk on bioassays. And so the first question is from Nadine, and she asked, as you showed, there have been substantial operational advances in vitro bioassays with significant improvements in controlled, consistent assay performance. Even if variations in dynamic range don't impact relative potency, should we ignore the wide swings in run-to-run performance of the reference standard when we have such a controlled, consistent in vitro bioassays though?
Oh, Nadine, another lady close to my heart. That's a great question, Nadine. And I think that gets to some of the regulators' concerns of, okay, is comparison to a reference standard hiding the fact that your assays could be wobbling all over the place? And there, for me, I think it is, we should not get away as bioassayists from staring at the data itself. Because sometimes, like you said, Nadine, we can get so computational and using automation that, you know, analysts can get away from looking at real data. You know, you should always have assay controls that will show variability over assay to assay. But I think you should be trending and looking at your curves from day to day, because what it could tell you is, even though you have all these great controls within the assay comparing to reference standard, if things start shifting too much, maybe your assay all of a sudden is going to crash. So I believe still looking at data helps you predict and look at how your assay is performing, even though this modern way of working and all the work we've done over the years to control assays helps you get accurate and precise assays on a more reasonable basis.
Okay, thank you for that. There are a couple questions on, maybe this will be the last one in the interest of time, on ready-to-plate cells. So Varnica Roy asked a question about, are there particular vendors you recommend, or do you prepare these in-house for the RTPs? And then also Stephanie Whipple had asked, are there certain cell lines that do not work well for ready-to-plate setups because they have low viability after thaw?
No, great couple of questions. So yes, there are actually vendors that will do that for you. So you know, when I was at Genentech, we did it in-house. When I was at Amgen, we did it in-house. But when I moved to AstraZeneca, we actually used a couple of companies that can do it, you know, that can do it for us. And in fact, when we were looking at product costing, because some of these companies do it on a large scale, they can actually do it cheaper than we could. So we could hand it over to them, and they could grow a ton of stuff very quickly because they have, you know, they can work with their procurement to have like large numbers of, you know, batches of, say, sera and that sort of stuff. So you can do both. As I say, it depends on how you'd like to do it. But there are companies out there that can do it for you. And absolutely, there are some cell lines that just don't like being ready-to-plate cells, because you have to have something that is robust enough that when you bring them out from the thawing, that you don't have to wait so long for them to recover. So, you know, there are some cell lines that it's just not practical. You can do ready-to-plate cells, but they're not ready to plate in a reasonable time frame, because you have to leave them in the plate for so long to grow up that it's kind of not worth it, and you're never 100% sure whether they're going to make it or not. So absolutely, you have to try it out to see how robust your cell line is to those sorts of conditions. So thank you.
Perfect, thank you. So I think once again, we would like to thank you, Tony, for Tony's a friend of Bioassays. He has been for a number of years. It's really a pleasure for us to hear your thoughts on bioassays in the current time frame that we're working in. And so I think at this point, I am going to hand this over to Mike Sadik to give some closing remarks.
Thank you very much, Jill. And again, Tony, thank you very much for a wonderful keynote speech talk. That was very interesting and kind of nice to see the world in retrospective through today. Thank you, everybody, for having attended this virtual conference this week. I just want to leave us with a couple of thoughts as we end the sessions. First off, thank you to everybody from the CAS organization who has helped to put this on, Julie and the rest. It's been the work of many, many people. And I want to really give my appreciation to all of those who made this possible, and also those of us, my colleagues on the scientific committee. This conference has definitely demonstrated there's a lot of sharing of knowledge and capacity building. There's a lot of innovation going on in the bioassay space. It's not a static area. It can go in cycles sometimes where you step back and move forward, and step back and move forward. But it is constantly moving forward as a whole. Therapeutic modalities continue to evolve, such as the complex monoclonals, the cell and gene therapy, and so forth. The bioassay community as a whole is addressing new and complex challenges by innovating novel and creative approaches, strategies, technologies, and solutions. So long as we continue to share so much with each other, the bioassay community continues to grow and get stronger and better. Many of us have worked in this field for a very long time, but we still keep learning from each other. In fact, it's my adage to myself that once I stop learning new things, it's time to quit because there's so much still left to be learned. And a conference like this makes that possible. So who attended the meetings this week? We had 137 registered attendees. Of those, over a third, 54, were first-time attendees. That's pretty cool. Number of different companies represented, 53. We had 6 different global regulatory agencies at this meeting. We had representation by 12 countries: Belgium, Canada, China, Denmark, Germany, Ireland, Japan, Netherlands, Sweden, Switzerland, UK, and the US. And we had 17 people as speakers and or panel members. So that was our total of personnel involvement. One thing I want to remind you is, please, as you get your emails with the surveys, please answer them. The feedback is very important to us on a number of levels. First off, it helps to build our symposia for the future. It also lets us know what worked well and what didn't work well at this meeting, so we can learn from that and improve future meetings. Ideally, meetings next year will have a much higher face-to-face content, but one never knows with all the COVID variants running around, so it all needs to be seen. Please get the evaluations sent back. They should be sent to you by today, but please complete the survey to share your feedback. I do want to take this moment to acknowledge all of the events that the CAS organization has, anywhere from CE to this week's Bioassay, the CMC Strategy Forum, the WCVP, the higher order structure meetings, cell and gene therapy products, mass spec, the AT Europe. So there's a lot of different forums. I do highly recommend that you log into the www.cas.org and see what all is available. More than 3,400 members participate in the CAS interest-based symposia and fora. And also, please save the date for next year, April 11th to 13th. Ideally, we'll all be in Gettysburg. Again, there's no guarantees, but that is certainly the desire and hope. And vaccines willing, we'll all be there. So I want to thank everybody for their participation in the symposia this week, in the conference, And I hope you have a very wonderful and healthy rest of this year. Hope to see you at a conference soon. Thanks very much.